So in this video, we're going to be thinking about the cytoskeleton, which is a really important component of how eukaryotic cells are organised. So if we think about a cell, so there's our cell, we've got the nucleus there. And remember that eukaryotic cells are 20 to 30 micrometers across. Okay, so that's actually quite a big gap. Or quite a big distance that things have to travel in within the eukaryotic cell. So you can imagine if we have a vesicle over here, let's say at point A, and it needs to get to point B over there. If we just rely on diffusion, that's pretty slow. So diffusion of that vesicle would be in the range of less than 0.1 micrometer per second, which is relatively slow. And also diffusion, the vesicle might not go in a straight line from A to B, but it might sort of wander all around the cell, so it's pretty slow and unorganised. But thankfully, within the cell, we have a network of filaments, which is called the cytoskeleton. And travel via the cytoskeleton gives us around about 1 micrometer per second. So it might take 30 seconds to get across the cell, which is much, much more useful than having transport in the range of minutes. So there is this bunch of different filaments. So we might have some radiating out like that, for example. Or we might have some filaments just under the membrane to help with the movement of membrane proteins. So we have these filaments inside the cell which effectively act as roads, effectively, to help the movement of components around the cell and to make it faster. So there are a few different processes within the cytoskeleton, but they all have the same basic architecture. So what you have is some sort of filament which is made of repeating proteins. So you've got the same protein and again and again and again. And what can happen is that filament can grow because it can have new proteins added at one end or the other. So we have new proteins come on and attach at the end. And that usually is directional. So we usually have what's called the plus end, where new proteins are added, and we might have a minus end where the filament is starting to decay. Okay? So we have some sort of structural filament, and then we have these little proteins called motor proteins, which walk along the filament, and I really do mean walk. So if we've got a little motor protein there, they all tend to have this structure. So they've kind of got 2 feet that come together, and then there's a central spike up there. So if I just colour in one foot in one colour, so what happens is that protein will then pick its feet up and place them one in front of the other, so that sort of swings across. So we've got this kind of walking motion along the filament. That motion requires the hydrolysis of ATP. So this is another way that the cell is using up ATP. And if you remember, the hydrolysis of ATP releases energy. And that energy is being used to power the movement. So we have these little proteins walking, literally walking along the filament like that, which if those proteins are then attached to something, so let's imagine now that our vesicle is attached to that motor protein, these motor proteins moving along will pull that vesicle, so the vesicle will move along the filament, in this case towards the positive end. So we have these filaments, and we have motor proteins, and the movement of the motor proteins is powered by ATP. And there's 3 systems of cytoskeleton in a eukaryotic cell. So the first one is sometimes called the microfilaments. And they are made of actin. We have the intermediate filaments, which we know a lot less about intermediate filaments. And then we have the microtubules on our three different systems. So we're going to think about two of these systems. We're going to think about the actin system and the microtubule system. Okay? So our first system is the microfilaments. And these are based on actin and myosin. So in this case, actin is the filament and myosin is the motor. Okay, so they always kind of go together, actin and myosin. So microfilaments, so if I just draw out again, so microfilaments are pretty small. Okay, so they're about 7 nanometers in diameter, and they're made up of just repeating units of actin proteins. So each individual protein is a monomer, and then we get this tube or this sort of filament structure forming. Okay, and as I say, at the end you can have new monomers added at the end, which will elongate the filament. The filament will usually be bound to some sort of protein to start with. So at the end of it, you might find a protein. It's called an ARP, and that's a nucleation site. So that kind of is a protein that gives the filament somewhere to start growing from. So it starts growing from here, it's growing in that direction, and we're adding new monomers on at the end there. So actin is the filament, and we have then our myosin motors. So again, they look like this, that sort of structure. And myosins, there are a load of different families of myosins. They're sort of related proteins. Some myosins go towards this elongating end, which we call the positive end, and some myosins go in that direction. So it depends which myosin you've got as to which direction it's going in. And typically, in a lot of cells, actin we mostly find is that network just underneath the cell membrane. It's not always there, but in a lot of cells, you've got a lot of actin just underneath the cell membrane to help organise membrane processes. So that's our actin and myosin system. The other system is the microtubules. So for microtubules, which I'll do in blue, we have tubulin is the filament protein. And we have two types of motors for microtubules. We have kinesins and dynein. And these are the motors. So microtubules are quite a bit bigger. So you've got tube structure like that, quite chunky tubes. And then you've got loads and loads and loads of individual tubulin proteins. I'll just draw the first few. And we have two types of tubulin. So we have alpha and beta tubulin. So some of these, they kind of alternate. You've got an alpha, then a beta, then an alpha and a beta. They're just slightly different in terms of their structure. So the filament itself is made of tubulin, and it's much thicker. It's about 25 nanometers in diameter. Again, you can have proteins being added. So these are new tubulins being added, at the plus end, so that would make this the minus end. And then for our motor proteins, so we've got two different types of motor proteins. So one are the kinesins, which always move to the positive end. So for microtubules, the proteins are a bit more directional, so kinesins always move to the positive end. Dyneins always move to the negative end. So we sort of split the directionality. For actin and myosin, there's different myosins and they can go in either direction. For microtubules, we split the motor protein, so kinesins go in one direction, dyneins go in the other direction. So it's the same structure. Again, we've got ATP being used in all of these systems. So it was also used by our myosins to power that energy, to power that movement rather. And our microtubules inside the cell are organised in this more sort of spider way. So there's a structure just off the nucleus called the centrosome, and that is a kind of microtubule organising structure from the cell. So the microtubules all start at the centrosome and then they kind of radiate around the cell. So that centrosome sits just off the nucleus, so we've got that radial pattern. Okay. So we can have these structures move, causing movement of vesicles within inside the cell, but what we can also have are sort of gross movement of entire cells using these systems. So there's two different systems. So one, we could think about the flagella. This is for eukaryotes. Bacterial flagella are a completely different thing and not made of the cytoskeleton at all. What I'm talking about is the sort of flagellum that you might get in a sperm cell. Okay, so you've got the sperm tail there, and then there's the head cell. So that tail is used to, obviously, for the sperm to swim towards the egg. And the cross-section of that tail is quite complicated. So that's cross-section through the tail. And there are loads and loads of microtubules inside there. Well, not loads and loads. There's a very predictable structure. There's what's called the 9+2 arrangement of microtubules. So we have round the edge 4, 5, 6, 7, 8, 9. We have 9 pairs of microtubules around the edge and 2 in the middle. So each one of those little circles is one of these microtubules here. Okay, and then what we have between them are a whole load of dynein proteins. So let's just highlight dynein in pink there. So dynein, we have dyneins kind of radiating out to connect all these different microtubules, and we also have dyneins connecting around the outside. So what we have here is the dyneins are sort of move the tubules in relation to each other. So they're stuck. They're sort of more stuck to the microtubules, but they're kind of moving those microtubules relative to each other. So just so with my pens, so we've got two. If we're looking down, there's two of them. There's some of my microtubules, and between them there'd be a dynein. So what the dyneins are doing are forcing the tubules to make that sort of motion in relative to each other. And that, across the whole length of the tail, means that you get that kind of swimming motion, so the whole cell moves. Another way that the cytoskeleton can underpin movement is in things like amoeba. So here, it was the dyneins that are doing the job, which obviously are using lots of ATP. In the amoeba, so you have cells that are, let's say we've got a cell sitting on a surface. there's a nucleus. And we have some sort of signal, so some sort of molecule in the environment. What can happen then is you can get the cytoskeleton selectively making new filaments in one direction in response to a stimulus. So in this case, it would be actin. So we might have the actin relatively evenly distributed through the cell to start with, but in response to this signal, what we end up happening is a whole new load of actin filaments form in response to that signal. So that's going to push the whole cell to move in that direction. So we've extended the cell on one side because we've had actin polymerization in response to a signal. That's then causing the cell to move in one direction. So amoeba, it's a movement based on the filaments. Flagella, it's a movement based on the motor proteins. But both of those are ways that these cytoskeleton proteins can cause whole cells to move, as well as vesicles or components within the cell.